Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einol. The so-called central nervous system consists of two parts, the brain and the spinal cord. The brain is confined inside our heads, while the spinal cord is the long, thin, tubular structure extending from our necks to the bottom of our backs. In humans, the spinal cord has about 200 million neurons. One of the jobs of these neurons is to generate rhythmic activity for movements such as walking or running, that is, to be so-called central pattern generators. One obvious question to ask is how the central pattern generators are constructed. Maybe there are certain neurons in the spinal cord that are oscillating by themselves, that sets up the rhythmic activity? Or maybe the rhythmic activity stems from network behavior, that is, from networks of neurons that are non-oscillating by themselves, but that end up oscillating due to the network dynamics. Today's guest, Henrik Lindén, has explored the second alternative. In a paper that came out in 2022, he and colleagues at the University of Copenhagen developed a network model accounting for rhythmic activity observed in the spinal cord of the turtle. In the podcast, we talk about the spinal cord in general, how the recordings in the turtle spinal cord were done in the lab in Copenhagen, how the data were analyzed, and how the computational model, a two-population network model with firing rate units, was developed based on previous ideas in the field, how the model compared with the experimental observations, and what implications the findings have for the overall question of the neural origin of the rhythmic activity set up by the central pattern generators in the spinal cord. After many years in academia, Henrik is now working for a pharmaceutical company doing computational modeling, and we also talk about his experience here. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media, or otherwise. Okay, here is Henrik. Well, welcome to the podcast, Henrik Lindén.
Thank you so much for having me.
Yeah, it's, of course, we know each other from you were sort of working as a PhD student,
Yes.
right? In our lab. So that's back in the days. But since then, you've you've done several things, but you also worked a lot on the spinal cord.
Yes.
So, and I guess that's the topic of today. I mean, lots of people, or more people, maybe work on cortex, and like especially computational neuroscientists. But there's also the spinal cord, which maybe then many computational neuroscientists are not quite sure. They've heard about it, but not quite sure what it is or where it is. So what is the spinal cord?
Yeah, it's true. I mean, the spinal cord is often neglected, right, in computational modeling of motor systems in general, and of course, in some cases that's fine. But the spinal cord in itself is also a very interesting piece of neural tissue, I think. Because it's really the sort of outermost layer, if you will, of the brain, of the central nervous system, and it's involved in all of our movements that we do. Of course, there are, as far as I understand, there are neurons in the motor cortex that will directly sort of synapse onto motor neurons, so directly control movements. But a lot of our movements are also sort of shaped and controlled by the spinal cord itself.
Is it
So
especially these this like rhythmic activities? That's sort of like the walking and swimming and things like this, which is, or at
yeah,
least swimming in animals. Mm.
so there is this notion of a central pattern generator, a CPG, and that's a label for sort of networks in the spinal cord that are able to produce rhythmic or other motor behaviors in isolation. So it was actually, I mean, it's actually a very old field. Spinal cord research has more than 100-year history. And in the beginning, there was a little bit of a controversy. So one important person is Brown, Graham Brown, I think his name is. He came up with the so-called half-center model.
So this was back in the.
That was published in 1911, actually.
Okay, so that's
So
quite
it's
early.
more than 100 years old. And it's a simple model of how the spinal cord can generate sort of an alternating rhythmic, alternating or rhythmic sequence.
Yeah, like in running, or like a horse running, or
Yeah,
lamprey
exactly.
swimming or something. But
So-
before we go into that, sort of
Yeah.
just in humans, I mean, we have the cerebral cortex, and then this covering sort of like some nuclei and the thalamus and all these things inside the cortex, and then the spinal cord sort of just that sort of sticks out from the bottom. It's like this long tube that goes into the the bottom of the back or something, or like it's like a meter or something, or
exactly.
that's
It
the
starts,
spinal cord.
you have the brain stem and then the spinal
Yeah,
cord.
so the brainstem, so the brainstem is sort of below the cerebral cortex and the nuclei.
Yeah.
And then after the brainstem, you go. Then it goes over to the spinal cord. Is that it?
Yeah.
Yeah.
That's correct. And it goes. I mean, in humans and other animals with vertebrae, it goes inside the vertebra, right? You have the spinal canal, and then inside of that is the spinal cord.
Yeah.
So
because it's sort of protected by this inside the bone structure.
Yeah.
Right. So that it doesn't.
It's inside the bones, so it's protected there, and then there is also a protective layer, like the cortex is protected by the dura and so on. The spinal cord also has this kind of layered
exactly
to protect the neurons and the fibers. So it's and then there is the cerebrospinal fluid that it's surrounding it also. So it's that's connected with the rest of the brain.
So in many ways, it's sort of like not so different from the cerebral cortex, except like the shape is sort of like long and tubular, or long cylinder or something, thin cylinder. Yeah.
And it has, I mean, like the cortex or the rest of the brain, it also has sort of gray matter, which is mostly cell bodies, and then white matter, which is mostly nerve fibers like axons, with or without myelin. So just
of course, if you have this, for example, that people would become paraplegic after say a car accident or motorcycle accident or something, then it's sort of like the spinal cord or the nerves in the spinal cord is cut, right? Is that a typical thing?
Yes. I think then it can be, I mean, it depends on the injury, of course, but I think often it's the nerve fibers that are cut or damaged. So the signal doesn't come through anymore. But I think the cells themselves can still be. I mean, it depends on the injury, I suppose, but they could still be sort of somewhat functioning, because there has been. I mean, that's one motivation to go into spinal cord research is of course that it happens that people get injuries, and then we want to know as much as possible about the spinal cord to be able to help these people. And there has been really cool recent, a rapid progress in the recent years in treating people with spinal cord injury or helping them by electrically stimulating the spinal cord.
Ah.
And that in some cases can actually restore at least partial movements.
Ah. Yeah, because you said when you said nerves are broken, that is because that could be so like the nerves is a collection, a bundle maybe of axons coming from the motor cortex.
Yes.
Yeah, so that's a while in contrast to sort of things happening in the spinal cord because they have their own sort of nerves sticking out of the spinal cord, right?
Yes, exactly. So there are axons within the spinal cord, and then very largely speaking, there are these fibers that make the communication, and then there is the gray matter, which is cell bodies. And if you look at a transection of the spinal cord, they are actually in a kind of a butterfly shape. So they have a certain, there is some kind of
For those of you looking at the video, you can see now Henrik making butterfly drawings. Everybody can envision a butterfly. I think that's even the listeners, yeah.
And then very largely speaking, there are motor neurons, which are sort of often bigger cells that send their axons onto the muscles. So the motor neurons, they directly control muscles, make the muscles contract. And then there are interneurons, which are not directly contacting muscles, but they sort of shape the signal that goes onto the motor neurons.
So you have like some interneurons, like doing the computation, and then you have some output motor neurons. Right?
Yeah,
which was the sense.
Actually, a majority of the neurons are interneurons. So the motor neurons are only a smaller fraction.
I mean, you worked for many years in the lab of, I think, Runeberg at the University of Copenhagen. And then, so he or they, or I guess or maybe you even participate in experiments. I don't know, or
a little bit.
A little bit.
I did.
Wow. Yeah.
I mean, it's kind of difficult to do experiments well, so I was happy to rely on other people's data in the end. But I did participate in some experiments
because I guess they focus a lot on the turtle.
Yeah,
that was like the main animal that was used or in the lab.
Yes. So when I started in the lab, it was still turtles. That was the main experimental animal, and that had been established, I think, by Jørgen Havnsgaard, who is a professor at the University of Copenhagen. But he wasn't the first in the world. Sort of, there was a researcher called Stein who established actually the turtle as an experimental animal, especially in spinal cord research.
Oh.
And there are
this was again back in the whatever long time ago.
That was in the 70s, I think, or 80s. Yeah.
And Havnsgaard, that was maybe 80s, 90s.
Yeah, he was doing a lot of research in the 80s and then on until recently. So there are some good reasons why the turtle is a good animal if you want to study the spinal cord.
Yeah.
And one is that, of course, if you want to measure from inside the spinal cord, it helps if it's stable. So the tricky part with motor neuroscience in general is that if you initiate motor activity, that means things start to move, right?
Yeah.
And then if you have an electrode inside the spinal cord or maybe in the vicinity, that gives a lot of noise. So it's a little bit complicated, right? To measure something that's walking around. I mean, of course, people do this nowadays. People do recordings in behaving animals.
I never thought about this, but of course, nothing moves in the visual cortex when you look at something.
Yeah.
But that's different for the motor system. Yeah.
So the turtle, of course, has the shell. It doesn't have the vertebra that we have, but it has a shell, and the spinal cord is inside in this shell. So the shell stabilizes the spinal cord.
Okay.
And then the other good thing about turtle is that it's actually at least the type of turtles that we used, which were kind of swimming turtles, they hibernate, and they can stay for a long time also underwater. So that means that their nervous system is very robust to sort of low oxygen levels and so on. So we used a preparation where, the turtle wasn't actually alive anymore, but the spinal cord was alive inside the shell. Now we could measure from that while we initiated so-called fictive movements. So it's not movement that you can see, but it's the activity inside the spinal cord resembles what would be there if it was alive and well.
Because I saw this paper that we're going to discuss a lot, and is this something that I think came out in 2021 or something, where you talk about the hind limb movement of this turtle, but it wasn't really then the hind limb moving; it was the part of the hind limb, the part of the spinal cord that had to do with making this hind limb move, for example, maybe in swimming conditions or something.
Exactly. So the behavior that we used was actually it's called a scratch behavior.
Okay.
So this you can easily see it in sort of in an alive turtle, but also if the turtle is not alive and the legs are still intact. So what happens is that if you tickle it a little bit around the hind limb, it has this scratch behavior where it tries to remove whatever is tickling it. And that's rhythmic. So it's
that's a little bit like we have two cats and they do this also.
Yeah.
There's like a scratching thing. They can see, they sit down and they sort of try to they when they scratch with the back leg, hind limb, as it would say here.
Yeah.
Yeah.
I mean, it's when we if I again, this is for the ones who have the video, but if you want to illustrate it, it's very hard to scratch with your leg. I mean, for us humans, we tend to scratch with our arms, right?
Yeah.
But these turtles and other animals can actually scratch with their
Are there even people who can scratch themselves with their legs? Maybe some really, like, they must be very flexible. Okay, so then, so
that's the behavior, and it's very stereotypical and it's very robust to. So that means you can do repetitions or trials.
So I think you mentioned that this I think when I asked you before this, that sort of like maybe this spinal cord of the turtle that you're talking about is like something like, or the piece of the spinal cord that has to do with this about 1 centimeter long, or like order of magnitude, something like that. It's not very important, just sort of, and maybe like 1 millimeter diameter or something, or thick,
something like that. So maybe the whole spinal cord is something like 7, 8, maybe 10 centimeters, depending on the size of the turtle. And then like in humans, it's sort of a very long tubular thing, but it's a little bit thicker in the lumbar region, like the region close to the hind limbs, and then a bit also thicker in the cervical region where the forelimbs in the turtle or our arms are located.
So I see.
The spinal cord has this kind of it's shaped.
So it's okay. So it's thicker actually down, actually close to you where your legs are attached.
Yeah.
And thinner up where your arms are attached. And that's
it's thinner in the middle, but then again a little bit.
Okay.
I mean, again, it's not something that you can see. It's not like a huge difference, but there is this. There are more neurons probably in the regions which are close to the limbs.
Okay. So this was like sort of like what's called the lumbar, part
so
of
it's the lumbar region. And then if you sort of look into one segment, there are maybe something like a couple of thousand motor neurons and maybe 15, 20,000 or 16,000 interneurons.
Yeah.
So it's still, I mean, in a relatively small segment, maybe only half a centimeter long, or a few millimeter long. So it's quite, still a lot of neurons in a sense.
Yeah, like 20,000 neurons or something in this order of magnitude. Yeah, in this and these interneurons you say are like they are both excitatory and inhibitory.
Yes.
Yeah.
So they are excitatory, and mainly then they are using glutamate as neurotransmitter, and then there are inhibitory neurons, which either use GABA or glycine as a neurotransmitter.
Yeah, so that's, I don't know, because we mainly hear about GABA when we talk about cortex. So maybe this glycine neurotransmitter is something special for the spinal cord, or is it maybe?
Yeah, my understanding is that it's the main inhibitory neurotransmitter in the spinal cord. But I don't know so much about it myself. I don't know why, if there's a reason for that. But
No, or whether it makes a big difference, or
I
Anyway.
know that you can block the glycine receptor by strychnine, so that's why strychnine is a very powerful poison, because then you inhibit all the breaks in the system and
so then it's just like recurrent some sense excitation or some excitation.
Yeah, so then you get overexcitation.
And then you get cramps and stuff. Yeah, so that's why strychnine is an example of a nerve poison.
Yes.
Yeah. And then this different, how much is, I mean, if you now look at sort of like cortex, for example, in a mouse, now we know, or in I guess in most, particularly mouse, maybe you have all these different interneuron types, right? At least I think you have 3 or 4 main types in the cortex. And how much is known about the types of I guess, interneurons and inhibitory neurons and
so there has been in the past maybe couple of decades, there has been intensive research, especially in the mouse, because there it's possible to look at genetic expression and so on. So there is many different types of neurons, or there is a classification of different neuron types, mainly based on the genetic profile, which is of course very useful to know, like if there are cells, where are they, and so on. So that's I think quite fascinating and useful to know about. But the link between genetic subtype and function is a little bit more blurred. So there are certain excitatory neuron types that are important for generating rhythms and so on. But if you compare our sort of conceptual models of the spinal cord with anatomical feature or functional feature, it's more blurred in terms of in reality, so to speak. Whereas the most modeling that has been done, it's more modular, where you sort of think of one neuron type having a specific role or function in the network.
So at least sort of the goal then for a modeler, as you were on this project, was sort of to understand what this spinal cord circuit is doing. So just to make sense, so that then we have this spinal cord circuit with this, say, like, some thousand neurons, excitatory and inhibitory, and then you also have this like motor neuron output, which really then the axons of those motor neurons becomes these nerves that is protruding from the spinal cord and going to the muscles. And so that's sort of the output in some sense. And then the input, what's the input to this circuit?
Yes, so in the if we look at the experiment that we try to model here, there is a sort of a sensory trigger to it, because as I said, you kind of tickle the turtle shell a little bit, and that gives probably an excitatory drive that lasts for a little time. So it's a sort of sensory initiated, but then and that's it. And then so there is we model it as a kind of a slow depolarization, but then the sort of rhythmic activity should come from the interneuron network itself. That's the way we set it up, and that's kind of the definition of a central pattern generator, is that it's capable of generating rhythmic behavior in isolation. And maybe, I mean, in many cases, I think that's it's an oversimplification, but so usually there will always be some sensory feedback, and and so on.
Yeah, and I guess
But
sort of is like you have some things that you do automatically, like when you're out running, for example, or whatever, or but then you have some kind of way to override to stop running, or so you have some kind of. But then you think of it more in this context as some kind of switch that tells it to turn on and turn off. It's not like
yeah,
it's not like
exactly.
something you sort of has to be going on continuously in order for this circuit to oscillate.
Yeah. So we set it up so that it should have a certain working point, if you will, the network that is different from a resting state. So there must be somehow a way to switch it on or off. And in our case, we thought of that as the sensory drive, maybe sort of that lifts or that pushes the network into a state where it will be active. Yeah.
So first, let's then look at the sort of like of course, the physiological measurements that were done in the group, right? So there was like you had like for one thing, you could measure the nerve output, the output from the circuit, something called electroneurogram.
Yes.
Yeah, what's electroneurogram?
So it's basically the summed activity that comes from all the summed activity into one nerve bundle that goes to one muscle, coming from the motor neurons.
So that's like a cable of axons from motor neurons.
Yeah, and then you measure the electrical activity kind of around it using this sort of suction electrodes.
Okay.
So it's kind of a crude measurement, which gives a very sort of it's actually a high frequency signal because you measure kind of a collection of action potentials that travel along the axons in that nerve bundle.
So then you can measure the output from these motor neurons. So I guess this is maybe like a technique they've had for a while because it's not so delicate.
That's an old and relatively simple, robust way of measuring the nerve output. So what you then see is that if you have different nerve bundles going to different muscles, and usually the muscles in a rhythmic behavior, they have this sort of flexor extensor division, the muscles, I mean. So you can see that the nerve signals that go to one muscle, the flexor, let's say, is out of phase with the extensor. So these signals are alternating.
Oh, exactly. So you can see that from the output is sort of alternating, like either one or the other, some sense, like so I say either the same with the left leg or right leg or something like that. If you sort of think of running or swimming, something like this. So okay, so and then what is then a more modern technique? It's sort of to measure the neurons, the interneurons, excited and inhibitor neurons inside the spinal cord with these multi electrodes.
Yes. So we used, or the lab uses extracellular electrodes, which are similar to the ones used in cortex, where you have this sort of multiple shanks with many or several electrode contacts on each shank. So just like people who measure in the cortex, we use the same type of silicone probes with a little bit larger spacing, but similar idea. So we could measure about maximally we use 256 channels. So eight
on each of these, on each of the probes, or had like several probes next to each other?
No. So it was let's say it was 8 by 8 shanks with 8 electrodes on each. And then, so that's 64. And then we had maximally 4 of these multi-electrodes at the same time.
Yeah. So what was do you remember the distance between these shanks? Because I mean, it's they like the whole thing. Yeah. So what was the distance between these, roughly, or maybe it didn't go Yeah. I'm just thinking, this should sort of, it's like a 1 millimeter diameters. So you would like the contacts you're interested in are probably within this 1 millimeter diameter, roughly like that, right?
Yeah. So the spacing,
not so important.
To check the methods, but I think it was something like 50 microns apart.
It's all in the all in the paper, and the paper is linked from the homepage of this episode. So that should be something one can look up.
But the important thing is that we could do spike sorting. So we could isolate individual neurons and assign action potentials to individual neurons. So we could measure a whole population at the same time and then look at their spiking activity.
Yeah, I saw that. Well, I think there were like 200 neurons in the paper.
Yeah.
So that gives like So then, of course, that's exciting. So what did this? If you just looked at the data, what did you see?
Yes. So we saw, of course, activity, and there. So what we typically saw was that there is a little bit of low frequency spontaneous activity in this preparation. But then if we initiate the scratch behavior, we would, at the same time as the nerve activity started to alternate, we would see activity in the interneuron population, where sort of one neuron was active after the other. So usually neurons, they give out more than one action potential during one sort of burst.
Cycle. Yeah.
And I should say the scratch behavior is relatively slow. It's about has a period of about a second or 2 seconds. So it's a slow wave kind of going through the network. So if you sort the neurons according to when they are active in the cycle, you could see that almost all phases of the cycle were represented or coincided with the firing activity of some neurons.
Yeah, exactly. So there was like this all. So this, I think you did some like analysis of this more formally using some principal components analysis, and show that this is what you call rotational dynamics. Right?
Yes.
So rotational. So the rotational dynamics rather than sort of alternating dynamics. Is that like So just like measure, this is an important point. So what is just like with alternating dynamics?
So often in the spinal cord literature, people assign neurons to sort of one distinct phase of the activity because it's many rhythmic movements, they have very distinct phases in the muscle activity. So it's sort of conventional to, if you find a neuron that is firing, you assign it to one or the other group of neurons. And that kind of goes into the way people often think about the spinal cord of being very modular, that some neurons are active in one phase, some other neurons are active in another phase.
So then you can have like two modules which are 180 degrees out of phase. Or whatever,
and of course, if you have many muscles involved that are a little bit out of phase to each other, there could also be more phases.
But still, that like in the simplest case, 180 degrees will get this alternating behavior as you see with swimming with different arms.
And that's not really what we saw. We saw more of a sort of continuous distribution. So it's based in the data we saw that it would be kind of a stretch to divide it into very distinct groups because it's more like a continuous wave of activity, if you like. If you then want to represent this wave of activity in a simpler way, you can do what you mentioned there. We did a principal components analysis, which is sort of a reduction of the dimensionality, and it turns out you can explain this population activity with very few principal components or very few dimensions. So if you project it onto 2 dimensions and you plot them one in a phase plane, you get something that looks like a circle, and that's sort of what we then call a rotation. And it's not something we came up with in this context, but it's sort of continuing the terminology that was established about 10 years ago, or a bit more, in the motor cortex, actually. because in the motor cortex, it has, if we go back a longer time, it has often been, thought of as having distinct populations with neurons that can control a specific movement direction and so on, so that you kind of, similar to receptive fields in visual cortex, people would sort of classify neurons in the motor cortex based on which, if we're talking about the movement of an arm, for instance, you can assign neurons that are more active if you go one direction compared to another direction.
Yeah.
And then about 10 years ago, there was a shift in perspective a little bit, where Mark Churchland and others, they looked at recordings of many neurons from the motor cortex, and they saw that they could actually, if they did something similar, if they did a dimensionality reduction, they could actually find not sort of distinct pools of neurons being active in a specific direction, but they could actually see that there was what they call a rotation in a sub space of the whole neural dynamics. So I think that shifted a little bit. That has shifted a little bit the understanding of motor cortex, is that you have sort of you have network dynamics that correlate with a specific movement, and these can be seen as a sort of, not a full circle, but a sort of something of half a circle or a quarter of a circle, something like that. So that has been called rotational dynamics in motor cortex, the primary motor cortex, and other motor regions.
So then you sort of thought of this, yeah. So then you, in some sense, had this idea developed in the context of motor cortex, that you could sort of see, well, it's the spinal cord. Of course, you think that maybe, I mean, why should the spinal cord be similar to motor cortex? That's not obvious, but that is sort of what you found there. So then you see these. Okay, so then you see these oscillations, right? And then, in principle, you could think that could sort of, even though it's a rotational dynamics, you could have these certain cells in there. Like, there are two options, I guess. One is that there's nothing special about it. All neurons are equal. It's just that the dynamics of the network is sort of what makes this rotational dynamics. That's what you pursued. Going back to that model, but there's also this other alternative, that it's not. It sort of is this some cellular sort of pacemaker cells that sort of that determines these oscillations, and so they have different cellular pacemaker cells in. But that is that is sort of like the two main IDs at the moment. And I guess I think you mentioned that this cellular pacemaker ID has been around for some time.
Yes, so that's the typical conventional way of thinking about the spinal cord, is that you have some populations of neurons that are responsible for the rhythm generation, and then there are other interneuron types that coordinate this rhythm across the different motor pools. And of course, that's it's a very logical way to think about the circuit. And it's also, it has definitely been found neurons that are rhythmogenic. So if we, in terms of the computational neuroscience or theoretical neuroscience literature, there is this excellent book by Isikiewicz, for instance, that deals with a lot of different types of neuron models, and you can easily set up a neuron model that gives many interesting sort of bursting behaviors and rhythmic behaviors in different sense. So that is, I would say, a very good idea. The difficulty when it comes to experimental validation is that it hasn't really. So it has been found excitatory neuron types that are necessary for normal functioning in the spinal cord, but as far as I know, it hasn't been found a single population that is solely responsible for the rhythm as of yet, but maybe tomorrow there will be. And then, of course, that's a very good way of thinking about it. But I think what we tried to do in our model, it was not to say that these ideas are wrong or anything like that. It was just to give sort of an alternative explanation, or thinking about what if if we really take it to the limit where different neurons, they don't have specific properties that give rise to their function or their role in the network, but we look at a network which is very simple, and could we still get activity
Make it
that's
simple
somehow
in the sense that it's only the very simple neurons, nothing special about the neurons, like
exactly very simple neurons, no intrinsic rhythmogenic properties, and very simple connectivity as well. So we just took the starting point was actually models that have been developed for, again, in cortex, where we take just assume a population of excitatory neurons, a population of inhibitory neurons, and then randomly connect them together. So no specific connectivity rule, except that it's random which neurons sends input to which. We just made sure that the number of excitatory neurons the number of excitatory incoming connections is the same as the number of incoming inhibitory connections. That's the only sort of.
But then you also, I mean, if you think of this, if you have like this paired, like that's sort of quite a motif that has been studied a lot in computational neuroscience, is this interconnected exciter and inhibitor populations. And I think, I mean, this one thing that has one type of models have sort of focused on spiking and neuron models, so like integrated fire type, and for example, and like it's been some very nice work like by Brunel, for example, and others, showing sort of how you get sort of quite rich and varied dynamics out of these two population spiking models, and they also explain why you are in this asynchronous irregular state where you have this irregular spiking. So that is sort of, it's also connected with this balanced ID and this balanced excitation inhibition, that on average the excitatory and inhibitory input to a neuron is the same. But you did not sort of consider this was like a different branch of this type of excitatory inhibitory. This was like firing rate models.
Yes, so we took the starting point in firing rate models that were already out in the literature. I mean, I'm a big fan of spiking network models, but I think in this context we could study the main mechanisms using a simple firing rate model. So that's the approach we took, and also because it was building on, as I said, previous models. So one model that inspired me a lot was the one that was just presented in a paper from 2014 by Hennikin and Fogels and Gassner, I think it is. That was actually a model of motor cortex, where they used firing rate models to explain how you can generate these sort of rotational dynamics that we talked about before in the motor cortex. And in that paper, they actually did both. So they first set up a simpler model, if you will, using firing rate neurons. And then they actually constructed a spiking network as well, using the same overall ideas, but they represented each firing rate unit with a small group of spiking neurons. And they show that they could get the same behavior in the network, or they get the same type of dynamics. So I think it's possible to also, in our case, to represent the units we have with spiking neuron models.
So, but okay, so so there, okay, so that, because a big thing, a very nice thing about when you formulate in terms of firing rates is that you can do this sort of like this, because you're after oscillations, right? You're after oscillation in the firing rate activity, and then with this firing rate formulation, you're able to sort of to a little bit more systematically look for this situation, right? Because using this, exploring the eigenvalues.
Yes. So the nice thing about using firing rate models is that you can if you are in a specific state, if you look at a specific level of depolarization, then you can linearize the network dynamics around that point.
Yeah, so then you think of sort of, you have these neurons, exciter inhibitory, and they also get some external input, right? So that they can sort of be at different depolarization levels before the neuron network dynamics kicks in some sense, right?
Yeah.
Mm-hmm.
So, and we have in our model, we use, again, a similar firing rate model that has been published before, but it's a nonlinear function. So it will have different slope depending on the depolarization level. So it
some
means
kind of sigmoidal relationship between the the input, the activity, membrane potential, whatever you, and the firing rate.
Yeah. So so that means that the gain, if you will, of the firing rate function, it changes a bit depending on the depolarization. And then what you can look for is the stability of this working point. And that is well established in dynamical systems theory. So the approach is you look at a working point, and then you look at small perturbations around that working point. And so the stability of that working point depends on the interaction in the network. And you can actually you can do this with very simple example in the theoretical neuroscience book by Diana Nabbott. There's a really nice example, I think, in the appendix, where they look at only one excitatory neuron or population and one inhibitory population, and then they analyze, depending on some of the properties of the connections, whether this simple two population model would be stable or not. And they actually show there as well that so you can analyze the eigenvalues of the system, and if all the eigenvalues in principle for a dynamical system, all the eigenvalues, the real part of the eigenvalue should be less than zero if the network should be stable. And you can translate that into a network of neurons where you have a specific connectivity matrix and you also have some intrinsic leak. So neurons are leaky, right? That gives a shift of 1 on the real axis. Then it corresponds to all the eigenvalues of the connectivity matrix need to be smaller than 1. And so the linear analysis of the system only tells you what happens up to that stability point. So if you make sure all the eigenvalues are smaller than 1, any perturbation you make to the system, that means any sort of little shift in any of the neurons, a firing rate, would just decay back to the baseline. So a boring sort of silent state. But then if some of the eigenvalues approach 1, you start getting very interesting behavior. So if
that sort of approaches instability.
It approaches instability, right? And there is a classical result from Sompolinsky and others that show that if you have a network which is infinitely large, so if you make it sort of in the thermodynamic limit where it's infinitely large and all the couplings are infinitely small, then this stability point corresponds to a transition into chaos, which is interesting, and that holds true also for spiking networks. That was shown by Van Vreeswijk and Sompolinsky. But of course, if you think about a motor network, it's not very useful to have a chaotic network because you want the network to produce the same thing every time, and a chaotic network by definition doesn't, right?
Yeah.
So coming back actually to, I think, an issue that you discussed with Sasha van Albada when she was in your podcast, is that the scaling properties, if you go towards infinity, the way you set it up, it will matter in some cases. So what we saw in simulation, so not theoretical results like Sampolinsky, but sort of in simulations, we see that if we take a network which is not infinitely large, but sort of a reasonable, a smaller size network, then this point of instability does not lead to a chaotic state directly, but it would lead to a state where you actually get oscillations or rhythmic activity that seems to be stable enough, at least.
Okay.
And
then you thought, aha, this is what we want.
Yes, because we wanted something that was rhythmic, that was producing this kind of wave of activity, and that was initiated or generated by the network itself. So the sort of key aspects that we saw in the data, we thought could actually be explained by this simple model of a balanced network, which is driven a little bit beyond its instability point. Because it shows sort of qualitatively similar properties as the data we saw. So that's,
So then it's important that there shouldn't be too many neurons in the in the circuit. Is that maybe, or maybe it's some kind of, maybe the approximation, something else breaks down. But at least you don't want to be in this infinite limit.
Yeah. Actually, I think it could. I mean, this is going beyond what is in the paper and so on. But there's an interesting paper coming from the Ostoich group in Paris that looked at what happens in this network if you add a little bit of adaptation. Then you can actually steer away somehow from the chaotic state. So that's one of the papers that inspired me also, and that we cite also in our paper. So in principle, I think you're right. You don't want the network to be too big, but there could be ways of steering away from chaos.
This is also, I mean, I think this is sort of this methodological point here, that you obviously have this nonlinear network, and then you're able to phrase an important question on the behavior of this nonlinear behavior, the nonlinear network, in terms of an analysis of a linearized model. And with the linear model, then with like many differential equations, many neurons connect together. Nevertheless, if it's linear, then it's sort of something we know how to do, a very large linear model. And then you look at these eigenvalues of this, like when you formulate this as a matrix in a matrix as a matrix problem. So that's it's very elegant, I think, to be able to go via this and to get all this qualitative insight from doing this analysis of these eigenvalues. So that was cool.
I mean, again, it's also one reason why we picked this model formulation is that, of course, this is what people have, used before. People like Larry Abbott and so on looked at eigenvalue distribution. So we could, we could piggyback on a lot of those results, of course.
So the book, of course, the appendix you referred to is in the book called Theoretical Neuroscience by Peter Ayan and Larry Abbott.
And then Larry Abbott has a nice paper from together with I think her name is Kanaka Ryan, where they looked at the eigenvalue distribution of a network with excitatory and inhibitory neurons. So they give a recipe of how to set the connectivity strength up to get a specific spectral radius. So that's from 2006. That's a very nice.
So that's something to keep in mind, sort of like this technique, if you want to understand these networks, which is, of course, typically very hard to understand. But
I mean, it's a typical, I would say the typical physicist approach, right? You take something difficult and that you don't understand, but then if you make it a little bit simpler, maybe you can understand some of it. And linearization is a very good trick in general.
But then you can go back, like they obviously did in this 2014 paper, and also make like a spiking model where you and I make like a more complicated model and see if the qualitative behavior survives.
Yeah.
Right. So it's so you can go back and check your approximation in some sense.
Yeah.
It's just that if you don't have this, if you didn't have this sort of like way to think about an eigenvalues too, I would think just look around in this parameter space of these spiking models. That's sort of like that's a quite tough thing to do.
Yeah.
Right. So you need some kind of way to think about it.
Yeah. I mean, In general, I like this approach of looking at the same phenomena in models of slightly different complexity.
I completely agree.
I mean, you and me, we also did this in one study, right? Where we have, that was something completely different. But I just like this
try
approach
to understand the local field potential generated by population. Exactly. Yeah.
You set up one model which is maybe more realistic, but also more complicated, and then you make another simpler model, and you try to understand the more complicated with the simpler one. I like that.
I think it's a general point that sort of if you think of sort of what would when you sort of try to understand a system with the spinal cord or the piso cortex, right, that you would like sort of models at different levels of sometimes simplistic coarse-grainedness. And it's a combination of these sort of like models, simple models where you can actually understand what's going on, and these more complicated models, which are often closer to easier to compare with experimental data and are closer, in some sense, to the underlying biology. And so if they all fit together, then we feel sort of so this is like a multi-scale understanding. Is that the word? It's not really scale, it's the same scale. It's like multi-coarse graining understanding or something like that. Yeah. Cool. So then you sort of found sort of how these with your model, that with this rather simple model and nothing particular about the neurons. I think you had like a model with like, I don't know, 200 neurons, I said, so and a 50% excitatory, 50% inhibitory. Even though I understand in these interneurons, it's a bit unclear how many are of each type.
Yes, I don't think it's known exactly, especially in the turtle and so on, how many. It's probably not 50-50, but I think it's also maybe not like 80-20 like in cortex, but maybe somewhere in between.
But presumably you can sort of, if you chose sort of like 60-40 or 70-30, you would just have to change the weight a little bit, and then you still get change the weight exactly. So like they do in the cortex, where they like compensate for this difference, the 80-20% difference between excitatory and inhibitory neurons by changing having stronger inhibitory weights inhibitory synaptic weights. So good. So then, okay. So then you found from this, it gives sort of like the, I mean, you find these rotational dynamics that you see in the experiment by having this like a network model, and we're close to instability, and then the amplitude, I guess, then how amplitude is given by the common gain, the amplitude of the activity. So what is meant by that, right? Wasn't that what you said?
Yes. Right? So the whether or not the network is active or not, it will be dependent on the depolarization. So you need to give it a bit of a lift or a drive to get it started. And then what we did, again inspired by others, who we were inspired by the paper from Stroud and Vogels and others from 2018.
Yeah, I think I'll add the sort of links to these papers in the podcast episode because we talk about them so much, and also the book of Diane and Abbott if you don't really already know about it.
So what they showed was that you can actually sculpt the activity of these motor networks, again, for motor cortical network models, by slightly changing the gain, meaning the slope of the firing rate function. So you can, in our model, you can do that by two different mechanisms. One is because the polarization will change the working point and then the gain. But we also
Because the
set
gain
up
is essentially the derivative
exactly
of the activation function.
Yeah. But we also assume that we could give an additional input that would alter the gain of the neuron. And then we saw that could actually be used to control the activity a bit in the network. So we could control it either to adjust the amplitude. If we just increase the gain to all the neurons a bit, that would also increase the amplitude of the oscillations a bit. And then, inspired by the paper from Stroud et al., we also looked at if there were some neurons that were actually had specific roles somehow, even though they are just the neurons themselves are identical, but they, depending on the way they randomly have been wired up, we saw that some were actually able to shift the frequency a little bit. So some neurons would actually make the whole thing go a little bit faster. Some neurons would make it a little bit slower. So then we said, okay, let's just take the ones that have the largest effect to speed up, and we call these like speed cells. And then we took some other cells and call them brake cells. And then we saw that if we drive them slightly, these two groups a little bit with an increased or a decreased gain, that would work to kind of shift the frequency a little bit. So again, in a sort of a targeted gain modulation in this model would also lead to the ability to shift frequency a little bit.
So is there a way to think about this? If you sort of have this, if this is like if I'm out running and I want to speed up, often I worry about speeding down actually, but say if I want to speed up, then what that means that I could send some signals from some kind of something in the cortex to this particular subset of neurons in the spinal cord, and then sort of regulate the speed by if you increase the gain, for example, or the working point or something, then you can actually sort of speed up that way. So it's a way to regulate sort of like the speed.
So and I think the important thing was also that we could, depending on how you target the population, you can change the amplitude independently of the speed. Because it's my understanding that in some of the more conventional model with sort of intrinsic bursting populations and so on, it's a little bit tricky to control amplitude and frequency independently.
I see.
Because if you think of a bursting population, that's something where you need to go into sort of a saturated state, and then the oscillation is broken by some kind of intrinsic adaptation or something like that. So it's hard to, control those properties differently. But it seems it's
because I mean, if I don't know, if you sort of have like if you sort of see like a cat again, right? They can they can walk, and they can of course walk fast. I guess that's sort of like but then they can also run in different ways, right? I guess that's like the difference. They call it, So like also with the horse, like different horse races, whether they sort of gallop or whatever, run, whatever. So is that also something that can be changed, or?
So
like the pattern, not the speed.
What we in this turtle preparation we have sort of limited. It's kind of limited what we can induce, but it's actually possible to induce two types of scratch behavior. One is a sort of a more rostral scratch, and one is more caudal. So one is sort of scratching towards as a sort of forward, and one behind the leg.
Okay.
So it depends where you scratch it. You can make this turtle either scratch. So it will try to scratch where you tickle it, but then it could either be moving the leg forward to scratch there, or moving it backward. And that actually requires a phase shift between the flexor and the extensor muscles.
Flexor and extensor. So that flexor are the ones that sort of make the muscle tighter, smaller. like the and extensor is the other way, right?
Yeah,
Or
so it depends on if you think about an arm, it's like this is flexing and this is extending.
again,
so it's
a beautiful demonstration for those who
for
have the
those who are
video.
yeah. So
Like
the
a
flexing
strong
is
arming Henrik here. Yeah, very good.
showing my biceps.
Exactly. Biceps. Exactly. Good.
Of course, even in this simple behavior, there are several muscle groups involved, but it would require you to phase shift a little bit.
And that can also be done.
We actually showed in the paper that it can also be done by sort of more specific gain modulation. If you give a specific patterned input to change the gain, you can change the network activity. You can change the rotation enough that it's possible to read off very different motor outputs. So in our model, we assume that motor neurons are represented just by a sort of a reading out of the interneuron population. And if you that requires setting up sort of also the weights to the readout in a specific way, but it's then possible to change the rotational dynamics in the interneural network enough so that it actually gives very different outputs.
Yeah, cool. So, I mean, I think this is a very nice study. And of course, one thing it does is sort of that this balanced excitation inhibition idea is sort of now, which was sort of developed, I guess, in cortex. And I get like one main motivation was to explain this very sort of noisy spiking behavior, which is sort of comes out of this balanced excitation inhibition idea. And then it was also shown in motor cortex that with this kind of balanced excitation inhibition network, you could also get it's like the different kinds of like oscillatory patterns in some sense, which could be in it's like the central pattern generator kind of patterns. And now what you're showing, I guess, here is one thing that you have this rotational dynamics. That's like an experimental facts, and then you have sort of also used this idea to explain that this could account for like this central pattern generator behavior in the spinal cord. So then of course it's the question sort of how much of, if you should sort of speculate, sort of how much of the behavior in this spinal cord is explained by this versus this alternative pacemaker cell ID.
Yeah, it's a very good questions. Like what do you do if you have two different types of models that are designed in very different ways, and they can both explain the same thing, and maybe one has some advantages in explaining a bit more in this direction, some has advantages in explaining something else. And I think I don't have a good answer. I think it's a matter of modeling philosophy in a sense that if you want to understand something biological phenomena or any physical phenomenon, how to pick the right model, right? And I mean, I would say the model which we presented in this paper, it's definitely wrong in some aspects. It doesn't have any structure in the connectivity. It doesn't have neuron types. When we know there is at least some structure in the connectivity, there are neuron types definitely. But I think a model can be, as it said, it can be wrong but still useful, right? So you can have a one model that illustrates a specific principle by simplifying it down to really just highlighting that principle. And I think that's what we try to do. And then if we want to have a very good and detailed model of the spinal cord, we would need to, merge the two somehow. So the way I think about it is that maybe this way of generating a rhythm or generating a sequence, it's part of what the spinal cord does, but there are I would guess certainly also specific populations that do specific things, and there is for sure there is also specific connections that bind the left and right part of the spinal cord, which we didn't include. So there is certainly sort of fibers that
because when you sort of run like when animals run 2-legged, 4-legged, it's always like this, the left and the right leg is out of phase, right? That's sort of,
and that's very strong, right? It's part of almost any type of rhythmic behavior.
Except when you're galloping, I guess, like horses or cats or whatever.
And some of the conventional, I would say, conventional models by Ilya Rybak and others, they can actually explain sort of the switch between different sort of galloping and walking and so on. So they are definitely successful in that.
But I guess one thing you can say is that if the only thing you observe is rotational dynamics, then you cannot. So then that can be explained by a network dynamics, right? Alone, if that's the only observation, right? So, of course, if you have that, could also be that this could also be get rotational dynamics with a pacemaker cell for all network for all I know. So you need, I guess, one thing you need. Well, if it's the case that the spinal cord either operates by one or the other principle, then you need more experiments to distinguish. But couldn't it also be that both are involved? I mean, it's because can it make it more robust to have, like, if the body has two mechanisms for getting these sort of like a function, because it's a very important function, right? Running is sort of important.
Yes.
Yeah.
I think there are definitely both network properties and sort of intrinsic cellular properties that go together. And I think, for instance, having like a firing rate adaptation that can help to stabilize the activity in general. And so I think the approach we took is to try to focus on network principles alone, because I think that's just very interesting to try to understand networks in general, and also to shift the approach of focusing on maybe specific modules or neuron types so much. But I mean, in the end, it's a mix of both. I think that if there are neuron types that are specialized in some way, I mean, there must be a reason why there is a specialization. Otherwise, it wouldn't have evolved, I guess.
No, that's true. Or maybe it's easier to make. No, I guess specialized cells. Depends on what is hard to make. Is it easier to make all very similar neurons, or compared to, I don't know. We leave that to the genes and the evolution to find out what is.
And then also the other aspect is that I think now we talk about the spinal cord, but of course the spinal cord is different in different animals and in humans, right? And even in simpler animals like crabs, isn't it? What's the name of this? The preparation that Eve Marder has studied extensively.
Gastric ganglion or something.
Exactly. And there are very few neurons that are known to have rhythmic properties, and that can, in that animal, that's enough. That works there. So I think it's also a matter of complexity, in the sense that if you go towards higher level animals, they could have slightly different principles.
Yeah, there's also this, at least there's this argument that if you sort of look at the number of neurons in really simple animals like C. elegans, I think it's 350 neurons or something, or maybe also this stomatogastric, which has at least very few neurons, that the number of neurons compared to the size of the DNA is it's such that maybe you could have more information in the DNA for each neuron in some sense. So there could be more information. But while, say, if you have, like, I don't know, in the human DNA is not that much larger than the crab DNA. I don't know, some orders of magnitude, but the number of neurons in our spinal cord, is no way that you could sort of, that's special. I mean, specify each of the synaptic weights in the human brain or the central nervous system just by the DNA. So it shouldn't be so that surprising if it's sort of some different principles for really small animals compared to let's say, humans or large mammals. Or
Yeah.
yeah. So I think that's, but it's interesting. So okay, so this, I think this has been really nice. So I'm very yeah, I learned a lot, and that's always fun. And I think it's a very nice example of sort of bringing together different pieces of already established theoretical insights that you could sort of pick from the shelf and put together and modify to explain a new system.
Yeah.
Yeah, that was really cool.
So, and actually this paper that we are talking about now, it was started already in 2013. And the good thing is about being slow is that other people published very interesting papers that we could learn from, and we could, take bits and pieces. And
Okay.
yeah, so that's
So is
been
that
good.
some advice to young investigators? If you're stuck, just be
Yeah,
very slow, and hopefully somebody comes out with a publication showing
exactly.
how to solve
Solves
your
the
problem.
problem.
Yeah.
So that helped a lot, actually.
That's good. Yeah, so that's cool. Yeah, but it really illustrates the importance of trying to follow what's going on in the field, and in particular like methods which are developed sort of like more generally, that you can use in sort of in a more applied setting, or to explain particular experimental data, like in your case.
And also, I guess to be open to other disciplines a little bit. I mean, it could be other brain regions, but also what's going on in other types of science, what's going on in network science, for completely different things. Maybe there are tools or ways of analyzing problems that you can use.
That's actually one of the motivation why I started this podcast, was exactly this, because it's maybe there are someone listening now and saying, this kind of rate model could be very useful for my problem, because it's very difficult to follow the literature. At least I'm not able to do that in any meaningful way. It's very sort of anecdotal and accidental what I sort of come over. So
yeah. And it's because it takes time also, right? To really read a paper, understand it, it takes a lot of time. So
yeah,
it's limited.
Getting like a 1-2 hour podcast, which you're able to sort of learn like the basics of what goes on. That's like a much more efficient and more pleasant, I think. I'd rather listen to podcasts than to read papers. I think it's just it's more fun because they can combine it with like a walk or whatever. So before we end this, I mean, you worked in academia for quite a few years as a postdoc and researcher, and for the last couple of years you have worked in. I mean, you're still in Copenhagen, and but you have worked for a pharmaceutical company, because Denmark, for all computational neuroscientists especially in Denmark, but maybe people who would like to move to Denmark, you have a very strong pharmaceutical industry in Denmark, which is very nice for the field. Being a Norwegian, living in Norway, I envy you all these pharmaceutical companies. So how was that? How has it been? You've been there for a couple of years. So what you worked on and so on?
I've done that for a couple of years now, and it's still I'm still doing science, doing research. It's just in a different context. So I really, enjoyed working in academia as well, but I also enjoy having a job. So
Permanent job.
permanent job.
Yeah.
So
And
so
pay
I
your
thought I
bills.
would
That's good.
see if there's what else is out there.
Yeah.
And I was positively surprised that I was positively surprised that I feel like I can use a lot of the approaches, a lot of the techniques that I've learned doing modeling in
Like
neuroscience,
solving large sets of differential equations, for example?
Exactly.
Or?
So I work with large systems biology models, trying to combine that with modeling
So what is a
pharmacology.
systems biology model? Systems biology is like not self-explanatory, because it just
Yeah,
from the name,
so
it could mean many things.
it could be explaining protein interactions, or trying to understand a specific disease, where you then try to model the different components using either modeling the different usually modeling the different reactions between proteins, between a drug and a specific pathway. That could be sort of intracellular dynamics, or it could be lots of different things depending on the again, level of detail and so on. But I work with what is called quantitative systems pharmacology, which is sort of bridging systems biology or OD-based models of biology with pharmacological interventions.
Exactly. So that's the idea, that you get models which can sort of give some hints or predict what kind of drugs may change the behavior and maybe cure a
exactly. I mean, it's I think anytime you have more than two things interacting is difficult to understand conceptually. So that's when you need, then when you can be helped by a mathematical formulation. And once you have, you have your interactions, you have your assumptions written down in equations, you can simulate that and try out different things and test, does it behave as we think it does? Are there maybe smarter ways of inhibiting a specific pathway than that we are interested in than we think? And can we also try to estimate what would be necessary of a drug to be effective enough? And that can be done sort of based on what is known about the biology. You can do that in computer simulations without having to try, ideally without having to try it in animals and so on. But of course, it's never either.
So do you go to like the traditional scientific conferences and publish, say, like scientific papers, or how is that culture?
Yeah, so that shifts a little bit, right? Where in academic research, your main outcome is publications, or maybe and presentation, and maybe teaching and so on. And where I'm now, the setting is more, so I support research projects where there is a specific focus. I try to work on develop models that can answer specific questions. And so far, it has only been presented internally. But in some cases, I haven't done that so far. But we also publish results and present at conferences.
So what about? I mean, it's like I guess there's like a lot of. It's not all machine learning, right? Still, I mean, you still do traditional differential equations and stuff, right?
Yeah. I mean, machine learning is definitely also a hot topic.
Also within the company,
data science and so on. Trying to get as much as possible out of existing data and also publish data and so on. But I think I really believe that there is a need for mechanistic modeling, even if you have powerful machine learning models. I think a simple and good formulation in sort of mechanistic terms can still be very useful to try out ideas, to try out, do we understand the system enough? And I mean, I think, sometimes I've heard this expression that a model is never better than the data it builds on. But I think that's very much shaped on the recent, machine learning type models. I think a model can be very useful without any data. If you are, I think maybe you could have trained an deep learning or trained a network to, infer like Newton's second law. But if you are just lying there and thinking a little bit, you can formulate it with very little data, right? I probably couldn't, but Newton could.
No, that's true, but still, it's sort of getting like this explainable AI. Even if you have a network that can predict sort of what happens, where the planets will be using AI, if you don't understand it's difficult to extract these sort of like this simple to formulate Newton's laws from just having a network, an AI network that can do this, right? Make the predictions. So that's different thing. But it's okay. So we certainly agree on this. But of course, we are biased. We are both sort of physicists in training, but also obviously the company appreciates this, right? And also the people who pay you.
Yeah.
So that's
No, but I think traditionally, like pharmaceutical companies, they have relied a lot on animal testing, right? And I think the general trend is that we want to move away from that as much as possible. So and rather use existing data, rather use computer simulations to the extent possible. Also, in some cases, animal testing is not really that good because animals are not humans, and so even if you test something in a mouse, a lot of drug projects, they fail because then it turns out it's very different in the human. So I think that's a general trend. It makes a lot of sense to try to establish modeling instead.
Cool. So I mean, this is sort of good because I mean, like, there's lots of people taking computational neuroscience PhDs, and you even went on to sort of to be postdocs and do research and so on.
Yeah.
And still, you found a rewarding career outside academia.
Yeah, I think it's.
So that's good to hear. So that should be. Yeah.
One should be open to different options, and I think there are definitely nice. There are good and bad things with everything,
Sure.
of course. But I think one shouldn't be afraid of going very deep into some academic subject, because you will learn a lot of tools. You will learn ways of solving problems that are applicable to other types of problems. You just have to,
Good.
find those problems later on. So
good.
So, yeah.
So on that happy note, it's always good to end with on a happy note. So thanks a lot for coming to the podcast, Henrik.
Thank you. This was very nice.